लिंक कुठे आहे इथे बरोबर हे वाला याला क्लिक करून कॉपी कर ठीक आहे ही वाली ना सेंड इथून ग्रुपवर कॉपी URL है ना ओके वेबिनार 2 है ना मैडम
वेबिनार
तुम्ही मुलांना सांगून ठेवलं आहे ना
मी प्रोजेक्ट करतो
इथून ना on this computer so very good morning Suresh sir
Good morning.
good morning one and all dear students participants and my colleagues this is the webinar series organized by SK Porwal College Kamti today the second webinar organized by Department of Zoology SK Porwal College Kamti is the part of this webinar series. I am Nitin Purushottam Beshram, Head, Department of Zoology, along with my colleague, Dr. Mrs. A.V. Ramteke Madam, Assistant Professor, Department of Zoology, on behalf of Dr. M.B. Bagde Sir, Principal, SK Porwal College Kamti, welcome you all in this August gathering. Today, we have with us resource person, Dr. Suresh Mishram Sir. Welcome, Professor Dr. Suresh Mishram Sir. In short introduction, Sir, working as Associate Professor in Postgraduate Teaching Department of Zoology, Rashtrasant Tukadoji Maharaj, Nagpur University, Nagpur. Sir also served as Deputy Director, Center for Bioresource Research and Management, Rashtrasant Tukadoji Maharaj, Nagpur University, Nagpur. Sir has published 33 research papers in national and international journal. Sir has completed one research project funded by UGC. Sir has 21 years of teaching experience and 10 years of research experience. Two students have been awarded PhD, three students submitted their thesis, and three students working under his supervision. I request Dr. Suresh Masram sir to start his lecture.
Thank you, sir, for my introduction. Today I am going to deliver on DNA sequencing. Nitin sir, I am audible?
Yes, sir, you are audible.
Now this DNA sequencing, I try to share the screen.
Sir, visible, your PPT. Your PPT is visible and you are audible also.
Thus, today we are going to discuss something about the DNA sequencing. Myself, Dr. Suresh Masram, Associate Professor, PGTD, Nagpur University, Nagpur. Now, this DNA sequencing actually it is to know what is the order of arrangement of different nucleotides along the phosphate and the sugar ribose backbone from either 3 prime or 5 prime. And this DNA sequencing, they are preceded by different processes, DNA isolation, then cutting of this long DNA into smaller fragment, then denaturation and multiplication of each fragment into the multiple forms. Now, before that, we again revise the DNA structure, although all of you well known about this DNA structure. The DNA structure, it has a two strand running from 5 to 3 prime, one strand and other strand which are aligned to it. It is running from 3 to 5 prime. Now this prime, they are decided by the deoxyribose sugar molecule, because in this sugar molecule from first, two, three, fourth, and fifth carbon atom, this deoxyribose sugar, it remains in this upright position, providing its hydroxyl group which are prevailing over the fifth carbon atom for alignment with uh, preceding phosphate molecule. And at last, here you can see this last deoxyribose molecule, it provides its hydroxyl group which are present over the third carbon. Now, and here is the adenine, 6 amino purine, cytosine adenine, always allied with the thymine by 2 nitrogen bond. Ribose, deoxyribose sugar aligned with nitrogenous bases by glycosidic bond, and consecutive deoxyribose, they are connected with each other by phosphodiester bond. Now, obviously, if we are interested to isolate the DNA from the animal, particularly animal, then this DNA, it is embedded in the cell embedded into the tissue embedded into the organ into the organ system, and organ system itself into the body. Thus, we have to do the biopsy, or if it is and take out the tissue, crush the tissue, and disintegrated the cells. And after disintegrating the cells, we have to disintegrate the different cellular part which are or which may attach us with this DNA molecule. Now, does this DNA, they are present in the cytoplasm, embedded into the cell, all these cell membranes, nuclear membranes, they are made up of the lipids, they are made up of proteins and different materials. Thus, first of all, this DNA must get rid of all these materials. And for this, we use proteinases to destroy the or to separate or to disintegrate the protein, type of detergent, surfactant to separate out the lipids. And then we also use the phenol to separate out uh, this intensely attached or intimately attached protein to the DNA. This intensely attached protein means histone protein are the proteins which are intimately associated with the DNA. They are separated by the phenol, and then the attachment of the phenol to the DNA, it is cleared by the chloroform, and again this proteinase enzymes or other proteinases, they are helpful to disintegrate or to separate out the protein from the DNA. Then the precipitations, they are carried out with the help of ethanol, particularly isopropanol, so that DNA should form the precipitate. Then precipitate of the DNA, they are purified by passing through mini column purification. Then comes the DNA technique. This DNA, which is very long DNA strand generally present inside the cell, from the bacteria to the higher stronger entity. And in sequencing, we could not sequence the long DNA fragment. It is not manageable to do this. Therefore, we have to cut this long strand into the smaller fragment and sequence each fragment independently. And after knowing the sequence of all these fragments, we realigned all these sequences and then could know the sequence of long DNA. Thus, the DNA, which is longer DNA, must be cut by the different distribution cases of the enzymes, and then each fragment go for the DNA sequencing. The next is the DNA denaturation. Double-stranded DNA is not used for sequencing. We have to denature it. The two strands of DNA, they are separated from each other by sodium hydroxide treatment and the heat treatment. And after that, we have to go for the PCR. Now, the single-stranded DNA is not sufficient to know the sequence of the DNA. Therefore, we must have the multiple identical copy of this single-stranded DNA fragment, and we could get these multiple copies of DNA fragment with the help of the polymerase chain reaction. Like this, thus, the long strand of DNA is not manageable. We have to cut this long DNA strand into several smaller fragments with the help of restriction enzymes. This restriction enzyme was shown by the scissor. The scissor or restriction enzymes, they cut this long strand into the smaller fragment. For example, if this DNA fragment is 100 base pair long, nowadays 100 base pair long DNA could be sequenced, but suppose we are at the stage of 78 or 80, where it is not possible to sequence the long strand of DNA, we could, and the desired DNA molecule, it is the 100 base pair long, then we have to cut it into the 10 pieces of 10 base pair long. Thus, A, B, C, D, E, F, G, H, I, up to A to I fragments, they are resulted after cutting the 100 base pair long DNA. Now, out of all this 10, then each one should be processed for the sequencing. Now, here, assume that this is the A fragment, and A fragment then must be denatured by providing sodium hydroxide treatment and uh, pitch treatment. After that, we receive the single-stranded DNA fragment. Again, remember, this single-stranded DNA fragment is not sufficient for the sequencing. Now, this DNA sequencing also involves the gel electrophoresis stage. Now, during this gel electrophoresis, to make this fragment visible over the gel or over the X-ray plate, because the gel is not used to exhibit the DNA fragment, but we have to take this DNA fragment from the gel to the X-ray plate, and then X-ray plate, they are processed for autoradiography, and during autoradiography, we could visualize the different bands which are earlier appearing uh, over the gel. Now, this tagging could be carried out on both ends, towards the 3' as well as towards the 5'. Now, the if we are interested to tag the 3' end, then we add the 32 phosphate radioactive molecule to the DNA strand with the help of phosphatidyltransferase enzymes. But generally, it is the 5' which are tagged by radioactive phosphate with the help of polynucleotide kinase. This polynucleotide kinase, it helps to add the radioactive phosphate, that is 32 phosphate, to the stranded DNA active. Okay, we go for the PCR. Now, during this PCR, the single stranded fragment, they are multiplied or they are amplified to in enormous proportion. During this process, single stranded DNA are put into the thermocycler. Then, to this thermocycler, we add everything which are needed for the replication. During the process of this replication, to carry out this process of replication, we add primer, we add DNA polymerase, we add different type of nucleotide. That means DAPP, deoxynitrosamine triphosphate, then GGTP, then GCTP, and DPTP. All these nucleotides, they are added to the reaction. Let's just now the polymerase which we are using into the PCR, it is specialized type of polymerase, that is top polymerase HU, top polymerase, which is derived from the hepaticus, and this polymerase it remain active at a very high temperature. At higher temperature, this polymerase it carried out the replication process, and therefore we use this kind of polymerase during this PCR. Now, when we add everything, a single stranded DNA it is multiplied. It undergoes the replication from the single stranded or double stranded DNA. After that certain period, that double stranded DNA again under the higher temperature and NaOH sodium hydroxide treatment, it gets denatured. Thus, we put the single stranded DNA, that single stranded DNA becomes double stranded DNA. That double stranded DNA undergoes the denaturation and give rise to single stranded DNA. Now, as everything which are already present inside the reaction mixture, which could carry out another cycle of the replication, both these resulting DNA strand, they undergo the replication and form the double stranded DNA. This double stranded DNA again undergo the another cycle of denaturation, and from this two double stranded DNA, four single stranded DNA are formed, and that each of these single stranded DNA undergo the replication and form another strand over them, and thus each single stranded DNA becomes double stranded DNA. Thus, with each cycle, there is multiplication or the amplification of the DNA from 1 to from 2, 4, from 4 to 8, 8 to 16. In this fashion, the number they are increases. Thus, with this with the help of this PCR, we could manages to have a multiple copy of DNA. Obviously, for the process of DNA sequencing, we required a single stranded DNA. Thus, up to the higher temperature stage, we allow the PCR, and because at higher temperature, there is the process of denaturation, and denaturation does it split all double stranded DNA into the single stranded DNA. We arrange the process of PCR and have all the single stranded DNA. Now, there are two main processes of the DNA sequencing. One is Sanger's method of sequencing. Another method is called as Maxson-Gilbert method of sequencing. Now, this Sanger's method of sequencing, this method is invented by Frederick 1977. During the same time, independently, other author Walter Gilbert and Alan Magazine. Alan Magazine was the student of is a student of Walter Gilbert, who is working under his supervision laboratory, and during their work, they published a paper in 1977, the new method of DNA sequencing, and during that paper, they describe their process of DNA sequencing. Now, in Sanger's method depends upon the ability of single-stranded DNA to undergo the replication. Thus, we have the idea that we must provide the DNA, single-stranded DNA, then allow the single-stranded DNA to undergo the replication by providing everything which are necessary for the process of the replication. After allowing the replication, but he manages to do something, to do certain thing by means of which it is assured that at certain point of time, at certain places, this replication must be arrested. And we should exactly know at which places that replication are arrested. Thus, he allows the replication of single-stranded DNA and manages to arrest this replication by doing certain trick, and that trick is the investigation of the new finding. And he also manages to make this, this DNA visible over the J or from J to the X-ray tape. Now, how he arrested DNA at the different places? Because when he added everything to undergo the replication, what he does, alongside all this material, he also adds a dideoxynucleotide. Now, this dideoxynucleotide is a molecule, a specialized molecule, which has hydrogen instead of hydroxyl group over the third carbon atom on deoxyribose. You know, in deoxyribose, the third and fifth carbon atom, they provide the hydroxyl group for the bonding. Now, as the hydroxyl group which are present over the third carbon atom, it is lacking there. Therefore, once such dideoxynucleotide attaches with the earlier or preceding nucleotide, it do not allow other nucleotide to get attached with it, because the extending hand which it may have in the form of hydroxyl group, that hydroxyl group is absent. Therefore, it cannot extend its hand for attaching with other coming nucleotides, and replication are arrested at that place. Thus, again, this is double-stranded DNA, and this is the normal nucleotide. This is ribose, this is phosphate group, and this is nitrogen. The adenine are shown, amino acid are shown. Thus, from second carbon atom, that deoxyribose allied with nitrogen is base. Then second carbon atom, it has hydrogen at both level, therefore it is not attached to other one. Now, at this third carbon atom, it has this hydroxyl group, OH group, which could attaches with other coming nucleotide. Because here at exactly at this place, there is a third carbon atom and this hydroxyl group. That hydroxyl group then attaches with the phosphate group, and thus uh, the phosphodiester bond they are formed here. Now, thus, if at this place instead of OH, if H are present there, then once this such kind of uh, nucleotide, dideoxynucleotide are attaching with the marching stand or marching DNA stand, then it is not allow attachment with other nucleotide, and thus if it is dideoxy adenosine triphosphate or ATP, then once that ATP attaches with the marching stand of we do not allow the attachment with other nucleotide, and the replication are arrested at the level of that dideoxy ATP. Now, thus we prepare the reaction mixture. We put all this reaction mixture into uh, into 4 centrifuge tube. In this each centrifuge tube, we add primer, we add DNA polymerase, we add deoxynucleotide, or four type of deoxynucleotide are placed. Thus, deoxyribonucleic triphosphate, deoxyribonucleic triphosphate, deoxyribonucleic triphosphate, deoxycytidine triphosphate, all they are added in each of these centrifuge tube or tubes. Now, in first reaction mixture, we add certain proportion of dideoxyadenosine triphosphate. Thus, in this reaction mixture, we assure that the replication must be stopped at the level of adenine. Once that dideoxyadenosine triphosphate touches with the marching stand, there is no further replication. Replication are arrested at that level. Thus, something happened with the second. Once dideoxy-GTP attaches with the marching stand of DNA, the replication are arrested at the level of 1. There is no further replication. Same with third, where replication are arrested at the cytosine level, and in fourth, replication are at thymine level. Now, after that, we go for the electrophoresis. Now, again remember, the DNA single stranded DNA fragment which we put into the all reaction mixtures, they are radioactively labeled by 32 phosphate towards their 5 prime. That is one thing. Now, we pour the content of this 4 reaction mixture into the first way. This 4 way, we pour the content of first reaction mixture into this first layer, and here we added the dideoxy ATP. Thus, we beforehand know that reaction mixture reaction, that is replication, are arrested at the level of adenine. Now, in second, we pour the content of second centrifuge tube, where we know that replication are arrested at the level of 1. In third, state of cytosine, and in third, the replication are state at the level of thymus. And here we show this different nucleotide by different colors. Now, where we write A, G, C, T, this top portion of chain, it is the area where we are forming the wave, and at these places we loaded the content of this secondary. From top side, or little side to the bottom side or positive side, during the electrophoresis, the DNA fragment, okay, now all the DNA fragments along this gel along this gel, they have entire in base pair single stranded DNA, and attaching with this single stranded DNA, there are another strand of DNA which are newly formed during the process of sorry, sorry, sorry. Thus, each strand now has an entire 10 base pair long single stranded DNA. Along with it, there present a newly formed strand of DNA which are formed in the course of replication. Now, thus, all the DNA fragments they have at least 10 base pair as the single stranded DNA which we put there, it is of 10 base pair long. Alongside, there may attach a single nucleotide during the course of replication because we put the dideoxynucleotide, and it is possible that the firstly formed nucleotide itself is dideoxynucleotide. Thus, once that a single dideoxynucleotide is attached with the primer, then they do not allow the attachment with the other nucleotide. Therefore, replication they are arrested here. Now, from top to the bottom, this different position of the DNA fragment, DNA band or the band, it indicates the molecular weight or length of the DNA. Thus, the lighter DNA, it runs very fast, and thus it could manages to reach the opposite pole. And the heaviest, it remained toward the side of the way because it will run very slow in comparison to the other. Thus, these DNA fragments, which are seen in the lane of the A or in the first lane, it first suggests that this is the lightest DNA. Lightest DNA, obviously, it must have a entire 10 base pair long DNA fragment which we see, and it must consist a single DNA, single nucleotide, and that nucleotide must be adenine. How we know that it is adenine? Because it is seen in the lane of the tube. Here we put the dideoxy ATP. Thus, here this blue strand, it indicates this our single stranded DNA fragment, and this purple color spot, it it shows the adenine. Thus, adenine get attaches with the with single stranded DNA. The next band from the bottom towards upward side, it is seen in the lane of G, or in the lane of second tube or second lane, where we put the dideoxy GTP. Thus, as we put the dideoxy GTP, we know that the dideoxy GTP once attaches, it does not allow the duplication. Thus, we know that this second nucleotide which are attaching here, it is the one as it is present in this plane, and it is next to the bottommost band. Therefore, it consists only two nucleotides. First nucleotide we beforehand know it is adenine because earlier band it is in the lane of A or in the first lane, showing that it must consist of adenine. Thus, second most is the guanine. Now, after this band, the upper band they are seen in the lane of T. In the lane of T. Thus, indicating that the third attaching dideoxynucleotide must be dideoxy and triphosphate, and therefore we analyze that the third attaching nucleotide is hydrogen. Next band, they are seen in the lane of the G, in the second lane. Thus, second lane, we know that duplication are arrested, must be arrested at the fourth nucleotide, and the fourth nucleotide which stop the replication, it must be the dideoxy 1 triphosphate. Therefore, we analyze that this fourth attaching nucleotide, it is the guanine, or the nitrogenous base is the guanine, and nucleotide attaching, it is the uh, 1 triphosphate. Next band, it is seen in the first plate. Here we from the bottommost, it is one. Therefore, we have to add the fifth nitrogen space here. Now it is seen in the lane of the A. Therefore, fifth nitrogenous base which are attaching here, it is adenine. Now same the next copper band, it is seen in the lane of cytosine. Therefore, the sixth nitrogenous base which is present in this newly formed DNA strand must be cytosine. Then just above it, there is again band in the same lane, indicating that the seventh nitrogenous base must be the cytosine, as the seventh nucleotide, it is dideoxycytosine triphosphate. Then above this seventh, this is the eighth band, and eighth band from the downward, and that eight bands are present in the lane of the tree, indicating that the eight nitrogenous base must be high as eight attaching nucleotide is dideoxy triphosphate. Above this eight, this is the ninth, which is seen in the third lane, where we add the dideoxy cyclene triphosphate. Thus, here we know or analyze that the nucleotide or the ninth nitrogen base must be high. Then the last, the tenth band, they are appeared in the second lane. Thus, attaching nucleotide is dideoxy guanosine triphosphate, and the nitrogenous base or the 10th nitrogen base is guanine. Now, thus, from this experiment, we have this red colored band indicating the single stranded DNA molecule which we put. Then we allow the replication. During the course of replication, we know what are the attaching nucleotides because we stop the replication at different places and in electrophoresis jail or when this jail are subjected to the radiography or we put the jail and X-ray film or we place the X-ray film over the gel and place such gel in the X-ray cassette. This is the X-ray cassette is a dark box, and that dark box is placed into the freezer in a frozen condition for a several days. And then whatever radioactive material which are present over the gel, they undergo the decay, and this decaying of this it form the marking over the area which are just over the X-ray film, and that X-ray film, when we subjected such X-ray film to the autoradiography, we could see these different tags. Now we mark the area where the veil are present. I now know which one is the proper side and downward side. That the area veil which has the veil, it indicated the area for the heavier DNA fragment, and the opposite pole it showed the area indicating the plane for lighter. Right? In this process of Sanders method, we put this DNA fragment, single stranded DNA fragment, and we allowing replication, we get know what are the nitrogenous bases which are formed during the scan. Thus here we know the sequences of newly formed DNA strand. That from the 5 prime, this sequence is A, G, T, G, A, C, C, G, C, G. Thus this 10 nitrogen bases they are present over this duplicating newly formed strand, and the DNA strand which we put, single stranded DNA strand which we put, it acts as a template. And obviously, as we know, A always attaches with the A and C always attaches with the G. From that, we compute the sequences of DNA fragment, single stranded DNA, which we are Thus, from this, we interpret the sequences of this fragment. We go for second, third, fourth, fifth, from B to the I fragment, and then again attaching the sequences of this 10 different fragments, we could know the sequence of longer DNA. And this method of DNA sequencing is also called as what? Deoxy method of sequencing, as well as chain termination of chain reaction or chain termination method of DNA. Then second method is Maxam-Gilbert method. This Maxam-Gilbert, Maxam Gilbert is the Walter Gilbert. Walter Gilbert is the professor in Harvard University. Was student when they attempt their investigation or their experiment in their paper, which is published in 1976. And method, it cut the single stranded DNA. And the manages to cut this DNA fragment at different locations. Specific cutting of the fragment, and then visualizing only one part of DNA fragment which are resulting. Then again we put all this into the electrophoresis, generally for the DNA. Otherwise, the electrophoresis are used, but for the DNA sequencing, polyacrylamide electrophoresis are. Now, this is the DNA fragment, single-stranded DNA fragment. Now, here there is ribosylglucose and there is guanosine or guanine, and this glycosidic bond of between the ribose and guanine, as well as deoxyribose and adenine, they could be cut with the help of formic acid. Thus, formic acid manages to cut the glycosidic bond which are present between deoxyribose and adenine, as well as deoxyribose and guanine. Thus, formic acid, if we put the formic acid into the reaction mixture, we can assure that the both guanine and adenine get separated from the DNA strand. Now, this separation of adenine and guanine is process is detylation. But if instead of formic acid we add dimethyl sulfate, then dimethyl sulfate most preferably it provides its additional sulfate to the guanine molecule, and after that it cut the glycosidic bond between guanine and deoxyribose. Thus, if we add dimethyl sulfate, then dimethyl sulfate only cut the guanine. Cut the guanine, separate the guanine molecule from the single-stranded DNA. Thus, for depurylation, that means to separate out the purines, we have two options. First, we can use the formic acid. Formic acid, but can separate both purines, guanine as well as adenine. But dimethyl sulfate, most preferably, typically, it also act on adenine, but which are present between the deoxyribose and one. Also, there is a pyrimidination. Now, this process is separation of the pyrimidine molecule, both with pyrimidine molecule, thymine and the cytosine from the single stranded DNA. Now, here, hydrogen molecule or hydrogen, it hydrolyzes the glycosidic bond between the deoxyribose and the cytosine, as well as between the deoxyribose and thymine. Thus, if we add hydrogen in the reaction mixture where we put the single-stranded DNA stack, then it would manage to separate out both type of pyrimidine, cytosine, and thymine. But if we add along with hydrogen, then in this condition, it could act only on the glycosidic bond which are present with deoxyribose and cytosine. Now, what is the significance of this depurination and depyrimidination? Once the purine here, once this purine or pyrimidine they are get separated from the DNA strand, then pyrimidine it manages to cut the phosphodiester bond. Thus, this cutting of phosphodiester bond by pyrimidine it cuts the single-stranded DNA into two fragments. Purpose of hydrazine, the purpose of dimethyl sulfate and formic acid is to depurinate or to separate the pyrimidine from the single-stranded DNA. But after depyrimidination, then there is the action of pyrimidine which observes such location where there is a no purine or pyrimidine, and at that same spot it cuts the phosphodiester bond and thus cut the single-stranded DNA. Thus, now again we put this single-stranded DNA into four reaction mixture into the four separate tubes. In first tube, we add the formic acid and again add the PCB. Now here the formic acid is depurinate or it separates the adenine and guanine from the nitrogen from the single-stranded DNA, and then pyrimidine it act on it and it cut the DNA fragment. As here, the cutting of DNA fragment they are at two places. Whenever there is adenine, it cut the DNA fragment. It also cut the fragment if there is presence of blood. Now, in second reaction mixture, in second centrifuge tube, instead of formic acid, we add the dimethyl sulfate. Now, dimethyl sulfate only depurinate or only separate the guanine, and then it will be it cut the DNA fragment between two fragments. Now, thus here cutting it is taking place only at the level of what guanine and not at the level of adenine. Now, in third reaction mixture, we add hydrazine. We add the hydrazine along with what? Only hydrazine, it separates out the cytosine as well as thymidine from the DNA strand, and pyrimidine, it cuts the depyrimidinated DNA fragment. In fourth, we add hydrazine along with the sodium chloride. If sodium chloride presents, the cytosine and non-thymine is also there. It carried out the cutting of single-stranded DNA fragment into two fragments, and here thus we assure that cutting are taking place at the level of cytosine only. Now, this is the single-stranded DNA. The fifth, 5 or 5 times they are radioactively labeled and typically are broken. This DNA fragment into two pieces. Now, when we put the content of this centrifuges into the way of electrophoresis gel, then what happens? There is a possibility, there is a presence of both this fragment, but only this fragment they are visualized, they can be seen, and they can interpret the position of, because when we pour the content, this fragment which is not radioactively labeled, they are also run along the chain, but they cannot show their presence over the X-ray film, and thus they are not analyzed and they are not meant to be analyzed. Now we come to the interpretation of this J electrophoresis. Firstly, we use J electrophoresis. Then again we put this J into the cassette where we place the X-ray film over the J, and we place such condition for several days in a dark condition, and then the subject is take out the cassette, take out the X-ray film, and subject such X-ray film to the autoradiography, and over the autoradiography we visualize different types. The same case here, whatever band which are toward the opposite side, it indicates that it is the smallest molecule. Now, obviously, smallest molecule must have what? A single nucleotide or single nitrogen base. Thus, here the lightest, again, the lightest band, it indicates the least number of nitrogen base. Least number is obviously, but thus here we show this adenine. Why? Because only the single nucleotide is attached, and there is no confusion because corresponding in the lane of G there is absence of any band. If there is corresponding band in the lane of G, then it means that this is the band of guanine and not band of adenine. But corresponding band is absent in the G lane of G or second lane. Therefore, we ascertain that it is the adenine. Then, second most position of band are this band. Now, in this case, in first lane we observe band, in second lane also we observe band. Now, the second band it can cut only at the level of the guanine. Therefore, this show that the second bond or the second nucleotide or the fragment which is 2 nucleotides long, it must have the second nucleotide as a guanosine fragment. Our second nitrogenous base must be guanine. Similarly, here in third lane also we observe the band. In fourth lane also we observe the band. It indicates that the DNA fragment, in the standard DNA fragment or open DNA fragment, it must have cytosine the problem. Here C, instead of A, we must read it as a C or a cytosine. This is C. Thus here, C, C are seeing a band are present here, band are present here. But in this, we know that the cutting, they are taking place at the sea level because both they are at the sea. Thus, this okay, so here this is the cytosine. The third molecule is the cytosine. Then a single band is present. Now, this band are present must be present here, thus indicating the fourth is thymine. Now, likewise, we go on adding or analyzing this uh, electrophoresis gel plate or X-ray plate, and we could analyze this this DNA fragment of sequential DNA fragment. Now, during this process, we analyze the sequencing of the strand which run from 3 to 5 prime. After analyzing the DNA fragment from 3 to 5 prime, we could understand the sequence of complementary strand which run from 5 to. Thank you very much. If you have any question, you can ask me.
Thank you.
Thank
you. Thank you.
Students, they are not they participating.
No, participants are there on YouTube streaming, sir.
Okay, okay.
So, thank you very much for giving such wonderful and elaborative lecture on DNA sequencing. Our students get really benefited with this, especially the students of third and fourth sem, that they're having this topic mentioned in their syllabus. So this will help them in the study and preparation of their examinations. So thank you very much, sir. Now I invite my colleague, Mrs. Dr. Iviram Madam, to deliver vote of thanks.
Thank you, sir. Good morning to one and all present here. Respected Principal Dr. M.B. Bakri, sir, Vice Principal Dr. Vinay Chauhan, sir, and Dr. Renu Tiwari, ma'am. Today guest speaker Dr. Suresh Masram, sir, Head of Zoology Department, Professor Nitin Majram, sir, and all the participants. It, it is a such honor for me to get the opportunity to bring a vote of thanks. On the behalf of the Zoology Department, SK Purwal College Company, I would like to express my gratitude to esteemed delegates of the webinar for their presence and contribution to make this webinar a great extent. I extend my gratitude to our today's guest speaker, Dr. Suresh Masram sir, to take out time from his busy schedule to grace the event. Thank you, sir, for your valuable and informative talk on the topic of DNA sequencing. I hope that students have been benefited from the webinar and should be implemented in the future. A special thanks to Dr. M.B. Bakri sir, Principal of SK Purwal College, for providing immense support to make the webinar successful. I extend my heartfelt gratitude to Vice Principal Dr. Vinay Chauhan sir and Dr. Renu Tiwari ma'am for their time-to-time support and help. I extend my gratitude to Professor Nitin Mishram sir, Head of Zoology Department, to organize such a wonderful webinar. I also extend my gratitude to Bhausar sir for their technical support. I congratulate to all the participants for their active participation. Thank you everyone once again for making it a great success. Now with the permission of chair, I declare that webinar is finished. Thank you. Thank you so much.
Thank you, Suresh sir.
Thank